welcome everybody. Chapter 1 of MCAT General Biology. Today's all about the cell. Chapters 1, 2, and 3 are really dense and high yield, especially chapter 1. And so a lot of this stuff might seem familiar to you, but there definitely are some stuff that you might not have even remembered or not even known. So definitely please pay attention, and you're most likely going to see a lot of this stuff on your test. So let's begin. We're going to start with the basics, okay? Let's talk about cell theory. There's 4 components to cell theory. The first one is all living things are made of cells. You don't really have to write this down, it's just like a intro. And so this alone already tells us why viruses are not living, because viruses are not made of cells, they're just made of genetic material surrounded by proteins. The next one is the cell is the most basic functional unit of life. And obviously cells have things inside of them called organelles, like little components that make them up. But the cell overall is the most basic unit that can actually do things, because organelles alone don't allow for life to occur. It's the compilation of organelles in the cell that allow for actual life. Now the third one is cells come from pre-existing cells. So you can't just have a cell come out of nowhere. This is talking about like replication or reproduction processes like mitosis, meiosis, binary fission, those things. And so one cell creates more and more cells, and those cells create more and more cells. That's what this is referring to. And the fourth one, cells carry genetic info via DNA. Emphasis on DNA, not RNA, right? Viruses have DNA, but some of them carry only RNA. Whereas living things, right, prokaryotes, eukaryotes, carry genetic info with only DNA. Obviously, we have RNA as well, but our genetic info mainly comes from DNA. And so there's 2 types of cells. We have prokaryotes and eukaryotes. So I'm going to compare and contrast them. Eukaryotes, right? Eu means true, and then pro means before. Karyote means nucleus. So eukaryotes have true nucleus, whereas prokaryotes, they don't have nucleuses, right? They have nuclei. They have something called a nucleoid, which is a region. The nucleus is an actual structure that has a membrane around it, and it carries the DNA, whereas nucleoid is just a region of concentrated genetic info. But it's not encapsulated by anything. It's not, membrane bound or anything like that. Okay, eukaryotes also have membrane bound organelles, whereas prokaryotes don't. So what's an example of a membrane bound organelle? We have rough ER, smooth ER, lysosomes, peroxisomes, but ribosomes, right? Ribosomes are not membrane bound organelles. This is something that both eukaryotes and prokaryotes have. But there still are differences. Eukaryotes have larger ribosomes, but L for large ribosomes, and prokaryotes have small ribosomes. And just in general, eukaryotes are larger overall than prokaryotes, and viruses are the smallest. So when we're talking about large ribosomes and small ribosomes, let's go a little more into detail. A ribosome is made up of 2 subunits, right? We have a large subunit and a small subunit. So in prokaryotes, the large subunit will be 50S. S is Svedberg units. So this is an S, capital S. And then the small subunit in prokaryotes will be 30S. And then together, what does this make overall? 80. That's incorrect. It's actually 70. Very weird, isn't it? So overall, this is going to make 70. Now, the reason for this is Svedberg units, they're not additive, and I don't really know exactly what the units are. It has to do with time, like sedimentation time, whatever that means, but they're not additive. So yes, 50 plus 30 is 80, but overall, it's actually 70 for, in this case. And then let's talk about eukaryotes. Eukaryotes will have 10 more of everything. So eukaryote large subunits will be 60 Svedberg units, and then their small subunit will be 40, giving us a total of
100.
No, it's not gonna be 100, it's gonna be 80. Same thing, it's really weird. You would think it's 100, but it's 80 in this case, overall. And how can you remember this? What I like to do is, I write this, right? Write in order, 30, 40, 50, 60, 70, 80. And this is all you need, okay? This first column is for prokaryotes, right? The ones with the odd numbers or the smaller overall numbers. And the right column with the even or the larger numbers are the eukaryotes, the ribosomes. And then once again, this is small, large, and overall size. So within prokaryotes, right, the main two prokaryotes we have are bacteria and archaea. Right, bacteria and archaea. Eukaryotes will be like, I don't know, other stuff, like pretty much everything else, like obviously animals, plants, we have protists, fungi, even like paramecium, amoeba. Prokaryotes are generally unicellular. I forgot to write that. They're generally unicellular, right? And eukaryotes are generally multicellular, but there are unicellular eukaryotes like those as I was mentioning, amoeba and paramecium. But that's a little too much detail. So bacteria and archaea, we're gonna compare these now. They're both prokaryotes, right? So they're both gonna have, no nucleus or just a nucleoid region. They're not gonna have membrane-bound organelles. But a big difference, right? Bacteria generally have cell walls, and archaea do not have cell walls. Another thing is bacterial ribosomes act like prokaryotic ribosomes. I'll elaborate on what this means. Give me 1 second. Whereas archaea, generally, their ribosomes act eukaryotically. So like, ribosomes, what do? They produce proteins, right? They allow for translation to occur. And so translation differs between both prokaryotes and eukaryotes. You don't have to know like too much about that, but just like if anybody has heard of like initiation factors, prokaryotes, when they do translation, they use something called IF, which is initiation factor, and then eukaryotes use EIF, which is eukaryotic initiation factor. So archaea, even though they're prokaryotes, they're going to use EIF, the eukaryotic initiation factors. Some of their ribosomal RNA sequences are more similar to eukaryotes than prokaryotes, and some of their proteins that they use to allow for translation are more similar to eukaryotes than prokaryotes. But the main thing I've seen that's been tested is just the cell wall thing, but everything else is pretty much same for both of them, prokaryotic wise. And then for eukaryotes, I'm gonna compare plants and animals. So obviously, plants have cell walls, and then animals don't. Plants also have a large central vacuole. Whereas animals, they have vacuoles, right? But they're not large and they're definitely not centered. And so the purpose of a vacuole is to, store water, right? So that makes sense why plants have large central vacuoles, because they deal with water a lot. Animals, obviously, they also need water, but it's not like as important as it is for plants. But something animals have that plants don't have is lysosome. And I will talk more about lysosomes afterwards, but like a general definition for lysosome is like a recycling center, right? They deal with like things that need to be recycled. And so if plants don't have lysosomes, what do they do to carry out lysosome functions? Do you have any idea?
Photosynthesize.
Not quite.
What did you say?
So lysosomes are present in animals, but not in plants. And lysosomes, they have functions, right? Like recycling and other things, autophagy, all that stuff. So if plants don't have lysosomes, what's gonna carry out lysosome functions?
Oh, in plants?
Yeah.
Being spade.
It's gonna be this large central vacuole. So vacuoles in general and animals, they just worry about like water and stuff. But plants, their vacuole acts as, of course, a vacuole, but also as a lysosome. And so the things that we talk about that the lysosome will do will be what the large central vacuole does in plants. And then finally, another difference is animals have a proper centrosome, which carries centrioles. And then plants don't. So both plants and animals have something called mTOCs. This is microtubule organizing center. Centrioles and centrosomes, they're like proper organelles that carry microtubules, whereas mTOCs are just regions that have microtubules. I don't know like too much in detail about those, but the main thing here is that animals will have centrioles, like proper centrioles, whereas plants will not. But they still both have mTOCs and they both have microtubules. It's just their organization is different. Okay. Now I'm gonna draw bacteria real quick. Okay. So this is like a general bacteria, right? These little dots right here are going to be our ribosomes. They're just freely floating, right? This right here, this big cluster is our nucleoid region. Remember, it's not membrane bound or anything. It's just simply a region of concentrated genetic info. These hairs right here are called fimbriae. And yes, they can help in movement, but that's not their main function. That's the function of this right here. But fimbriae are kind of like an attachment site for substrates or like receptors, you know? So they allow for like things to bind. And then obviously they have like cell walls and like they can also have a capsule, which is like an outer jelly type coating. But let's look at this right here. This was the flagella, but I'm gonna draw it better. So this overall is the flagella. I'm trying to break it up into two parts. Or three parts. This is also pretty important. Anybody know what this is called, like any of these three regions within a flagella? So we have the, let's try it over here. Basal body. Think about body is like the body right inside. So the basal body is inside part of the flagella. And then this middle part is the hook. And then the outer part is the filament. So, the basal body, think of it like a motor. The motor is going to generate lots of torque to allow for the filament to work, right? The hook is an attachment site. It connects the basal body to the filament. It's kind of like a joint, allows for free movement, free rotation. Now the filament, okay? The filament is the actual part that moves. And here's the thing, right? Prokaryotes and eukaryotes both have flagella, but the way they move is different. So for prokaryotes, they're going to move like this, like in a propeller type circular motion. And so in this case, when the torque is generated in the basal body, the filament will be able to propel. But in eukaryotes, the flagella moves like this side to side. Yo, can you check if I have space on this side? I always ask this for
You have no permission.
like all of this?
Yeah.
Okay.
Hey, so
Yeah.
like it'll literally give us a question like what are the three parts of the flagella?
I don't know. They might ask you that. I haven't like seen specifically what you asked, but they could. But at least like know like what the three parts do. Like they might ask you like, or maybe they bring up in a passage like this part was removed and so now the torque is like missing, something like that. And when you hear torque or like motor type things, that would be the basal body. If they're asking about the part that actually moves, you would know it's the filament.
Okay.
Yeah, I haven't really seen personally though a question like this, but so definitely could show up. All right, so next, we're gonna talk about eukaryotic organelles. Okay, I'm gonna start with the plasma membrane. So plasma membrane, right? The plasma membrane is made up of phospholipid bilayer, right? So a phospholipid, a single phospholipid looks like this. You have your polar head and then two like fatty acid tails. So bilayer, right? One layer would just be a layer of all this. And then bilayer means there's two. And so this could be like the inside of the cell and this could be outside of the cell. But the inner region is like non-polar, whereas the outermost regions are polar. And a feature about the plasma membrane is that they're selectively permeable. We definitely hear that a lot, especially in our classes. And that means that they can allow movement of things in and out of the cell, but obviously only certain things, right? So what can enter and exit the cell? Small and non-polar things. But how do polar or charged or large things enter the cell? We have things called integral proteins. So proteins that span the entire length of it. And these will allow movement of things, right? So water is a good example. Water is small, but it looks like this. And this is polar. So even though it's small, it's polar. It can't just pass through because of this non-polar region. And so a good example, right, is this aquaporin, right? Aquaporin is just a membrane channel that allows for water to come in or exit. And then in chapter 4 for nervous system, when we talk about that, we're going to talk about sodium and potassium leak channels, right? These are also small as hell, but like they're charged. They have positive charges on them. So they can't freely pass through. They need some sort of protein to help. Next is nucleus, right? Nucleus obviously carries the genetic info. And it's also the, what is the genetic info, guys? Is it RNA or DNA? Both. Yes, DNA. DNA. And this is the site of transcription. But it's transcription in eukaryotes. Where does transcription occur in prokaryotes if they don't have a nucleus? Or do they have a nucleus?
Cytoplasm.
Yeah, just a cytoplasm. Because nucleoid is just a region in the cytoplasm, so there's no proper organelle for this to occur. So this is transcription in the nucleus for eukaryotes. Remember, we're just talking about eukaryotes right here. Now, in the nucleus, there's a region called the nucleolus. People always forget about this one. The nucleolus is where ribosomes are produced via ribosomal RNA. Now let's talk about ribosomes. Remember, what are the sizes of ribosomes in the eukaryotes without looking at the board? These are going to be the larger ones, so 60 and 40 to make up 80. So what do ribosomes do? They produce proteins. But ribosomes are found in two main locations. One is, let me draw a cell. One is just in the cytosol, right? So you can have just ribosomes floating around, or you can have them inside of this endoplasmic reticulum, which is studded with ribosomes. And they do different functions depending on their location. So if a ribosome was in the cytosol, it's going to produce proteins for the cell itself, not including the plasma membrane. So it's just going to be for the cell itself. So maybe like motor proteins, right? Like kinesin and dynein. I'll elaborate on those when we talk about cytoskeleton. But you know, motor proteins, kinesin and dynein, will be made by the cytosolic ribosomes. Now, if we're talking about ribosomes in the rough ER, this will make proteins that are meant to be exocytosed, right? To exit the cell, so for things that are happening outside of the cell. And also these integral proteins for the plasma membrane. Okay. Now let's talk about rough ER, since we were mentioning that. The rough ER, or rough endoplasmic reticulum, obviously has ribosomes. Not much to say about it. It just helps facilitate protein production. And once again, it's for exocytosis or the membrane. But the rough ER is going to essentially, the rough ER, once it makes the proteins, is going to send the protein to the Golgi apparatus, right? Which will be the next thing I'm going to mention. But there's like an order that you need to remember for this. So rough ER sends it to the cis, then the medial, then the trans Golgi apparatus.
And it's sending ribosomes. Is that what you said?
No, it's going to be sending proteins that it made with the ribosomes.
Okay.
So this rough ER will make proteins and like maybe fold them and stuff, and then it's sending it to the Golgi apparatus. And so the Golgi apparatus, what this will do is post-translational modification. What does translation mean? Translation means like production of proteins, right? Transcription is DNA to mRNA, whereas translation is mRNA to proteins. So post-translational, right? After the protein is made, it's going to modify it. So this is talking about things like methylation, acetylation, and I don't know, oligomerization, those type of things. And then it's also going to package it up, the protein, package up the final protein, and allow it to be either exocytosed or sent to the plasma membrane. And so it packages them up in secretory vesicles. Okay, what else do we got? Let's talk about smooth ER next. So rough ER makes proteins, but smooth ER makes lipids. Also, smooth ER is a detox center. There's two organelles that detox, but detox can also make lipids. So those phospholipids that we have up there, where do they come from? The smooth ER. And one more thing that people don't often know is that smooth ER is a calcium storage organelle. Do you guys remember sarcoplasmic reticulum? It's a musculoskeletal. Do you remember?
Vaguely, yeah.
Okay, it's with muscle cells and stuff, but we're gonna cover that in the musculoskeletal system chapter. But the sarcoplasmic reticulum is just a muscle cell version of SER, the smooth endoplasmic reticulum. And so it's good to make that connection that the smooth ER is what the sarcoplasmic reticulum is based off of. And calcium is important for like muscle contraction and stuff, right? So the smooth ER will store calcium. Now let's talk about lysosomes. Lysosome, as I was mentioning, it's like a recycle center. So, maybe the Golgi apparatus is doing something, like it's modifying the proteins, and then there's some like waste that needs to get rid of. So lysosome will deal with that. This also does phagocytosis, which if you have like a bacteria or something that entered the cell, it's just going to engulf it, get rid of it, destroy it. And okay, another important thing, right? Lysosomes will do autolysis and autophagy. So what is autolysis, okay? Autolysis, think about like if you fell and you scraped your knee, you probably damaged your cell somehow, but your cell is still alive, but it's just like, it's not gonna function properly, it's gonna malfunction, but it's still there, it's just a burden on the rest of the cells. So autolysis means that it's going to essentially allow for that program to just die, right? It didn't want it to happen, but it has to because you fell and you scraped your knee. Autophagy is kind of like apoptosis, which is like, I have to mention apoptosis after, but apoptosis, right? Apoptosis means programmed cell death. So I'll elaborate on that, but autophagy is kind of like apoptosis, but for organelles. So maybe your mitochondria is like tweaking or something, or your lysosome is going crazy. So autophagy means it's going to get rid of the organelle and replace it. without causing too much of an issue to everything else. Okay, you know what? Let me just mention mitochondria then. All right, mitochondria, of course, we all know what it does, but mitochondria, to elaborate on this, is where apoptosis is facilitated. So apoptosis, as I was mentioning, is programmed cell death, right? Programmed cell death, meaning like maybe the DNA in the cell is like going crazy or something, or you guys know about T cells in the immune system. The T cells, like there's like a whole process for them to be selected. If they're not doing their job properly, then there's no use for them, so they get killed, right? Programmed cell death. And it's a very smooth, simple, process that's not messy at all. It just kills that certain cell, and it doesn't cause any problem to other cells. So apoptosis is chill. Autolysis is also kind of chill, but it's like, it's not really wanted, but it has to occur because of external reasons. And then autophagy is like apoptosis, but for specific organelles. And there's one more thing called necrosis. So necrosis is like a step further from autolysis, where like, let's say you really fell and scraped your knee like super hard. That might have just killed the cell alone. If you're falling, like these external reasons are actually killing the cell, then that's called necrosis. So nothing facilitates that. It just happens like based on these circumstances, like you fell, right? And this apoptosis, do you know why does the mitochondria, the one that allows for apoptosis, what does the mitochondria have that allows apoptosis to occur? It has to do with the electron transport chain. I think someone said cytochrome C. Yeah, so cytochrome C, right? This is part of the mitochondria. Don't worry, I will elaborate on where exactly this is. But cytochrome C, it's part of the intermembrane space of the mitochondria. Once again, I will explain what this IMS means. But it's part of the intermembrane space, and basically it gets released from the mitochondria, and then it activates this caspase cascade, which is just like a set of proteins. And then they all work together to allow for apoptosis to occur. But definitely know that apoptosis occurs because of the mitochondria, and it has to do with cytochrome C and caspase proteins. All right, so on that note of mitochondria, let me draw out a mitochondria. So this mitochondria is double membrane, okay? This internal part is this outer layer right here of the internal part is the inner mitochondrial membrane. If you go inside the membrane, this is the mitochondrial matrix. Now this outer membrane is just called the outer mitochondrial membrane, called OMM. And then the space in between the two membranes is the intermembrane space, or I call it IMS. And so the matrix is where your processes like the Krebs cycle, right, or beta oxidation occur. The inner mitochondrial membrane is where the electron transport chain occurs. And then intermembrane space is where the protons get pumped from the electron transport chain. And also parts of the electron transport chain, if you want to consider it that, the cytochrome C also resides here. And then outer mitochondrial membrane, nothing significant about that. But basically the cytochrome C is like sitting over here, and then just leaves, right? And then it causes that whole cascade to occur. So yeah, that's another thing I just mentioned, right? Mitochondria, apoptosis. Now for what mitochondria actually does, right? Mitochondria, we all call it the powerhouse of the cell. Why? Because it produces most of the ATP, right? Can you see the very bottom of this in the video?
Yeah.
Okay. Why do I say most ATP? Where else can we get ATP from? Or did I say it wrong? We just only get ATP from the mitochondria. Like what specifically? What? Yeah. So mitochondria will do all the stuff, right? Like electron, TCA cycle, electron transport chain, and then oxidative phosphorylation. That obviously does produce most of the ATP, like I'd say 99%, but we can also get ATP from glycolysis, which occurs in the cytosol. But it's very, very minimal amounts. Okay, so let me write here, ETC TCA cycle, and beta oxidation. Beta oxidation, for anyone who doesn't know, is just fatty acid oxidation. But beta oxidation does not always or only occur in the mitochondria. There's another organelle called the peroxisome. The peroxisome deals with handling like reactive oxygen species, right? So like this right here. This is a free radical hydroxyl, right? And so when it deals with reactive oxygen species, it generally produces this, which is hydrogen peroxide, right? Because when you have two of these radicals, they come together and form this, which is hydrogen peroxide or H2O2. And as I was mentioning, right, peroxisomes will also perform beta oxidation under certain conditions, okay? So beta oxidation is basically breaking up fatty acids, right? What is a fatty acid? It looks like this. Like just a general idea, right? I suck at it. Okay, it just keeps going, right? These carbons, right? When they're super long, like 22 or 20 plus carbons, we call them VLCFA, which is very long chain fatty acids. And so this stuff will not fit in the mitochondria, so you can't do beta oxidation on this. So the peroxisome will deal with the very long chain fatty acids until they become medium chain fatty acid or small chain fatty acid, which is like, I'd say like 10 carbons and below, okay? And once we get a 10 carbon fatty acid, right, whatever, then it will go into the mitochondria and it will perform the rest of beta oxidation. But the peroxisome, when it does beta oxidation, it does not produce any ATP. It will just mainly produce hydrogen peroxide. The mitochondria is where beta oxidation actually leads to ATP. It doesn't produce ATP on its own, but it produces the precursors for ATP to actually be produced. And then centrioles, once again, these are only in the, animals, not plants. But centrioles are just like a region, they like carry these microtubules and stuff, and they primarily help with spindle fiber formation. So like next chapter when we talk about mitosis and stuff, it's gonna be these things, right? That hold the chromosomes like in the center, like a metaphase, and then pull them apart, anaphase. That's like what the centrioles are used for. They're mainly like microtubule organizing center. Am I missing any organelles? Okay, I mean, obviously, there's vacuoles, right? I'll just write it one more time. Vacuoles. They store water, right? But what did I say is special to know about vacuoles? Is that vacuoles in plants are large and central, and they will take over the lysosome functions in plants because plants don't have lysosomes. If I missed anything. Yo, I actually cooked. Okay, I mean, obviously, okay, once again, there's like cell walls and stuff too, right? I'm gonna mention these are very, very basic, but like cell walls, what are they? They're just cell walls, right? They're just like an outer protection layer, only in plants, not animals. And then we have, what was the other one? Oh, I forgot. Yeah, okay, chloroplasts, also really small. The MCAT doesn't really talk about plants a lot, that's why I'll be forgetting, but chloroplasts are only present in plants, not animals. And so chloroplasts will do photosynthesis, which is converting light into energy. Do plants have mitochondria or just chloroplasts?
Don't they have a little bit of like some mitochondria, but the chloroplasts primarily take over?
Yeah, that's correct.
They get nutrients from the soil.
Yeah, that's correct. So yeah, plants will have both chloroplasts and mitochondria, but they primarily use chloroplasts. Animals just have the mitochondria, not chloroplasts. And then chloroplasts have chlorophyll, which gives the color to them, like the green color. Adding on to chloroplasts and mitochondria, there's something called the endosymbiotic theory. And this is like the theory that prokaryotes, like, okay, at first they were just prokaryotes, not like any eukaryotes. And so one day a prokaryote encountered a prokaryote and it engulfed it, right? And it became this prokaryote with another prokaryote inside. And this prokaryote inside ended up becoming the mitochondria or chloroplast. Like there's two scenarios, right? Chloroplast and mitochondria. And the reason this theory even exists is because, this is important, the chloroplast and mitochondria have their own what? Anybody know? They have their own DNA. So when we talk about mitochondrial DNA, we like read it as mtDNA. You'll definitely see these in some passages and stuff. But mitochondria are like the only organelle other than the nucleus that have their own DNA, right? I mean, the nucleus just carries DNA. It's for the nucleus, but it's the DNA for the whole organism. But the mitochondria has DNA for itself, like for mitochondria. And what does it do? Same with the chloroplast. They just allow for coding like proteins and stuff that are used in mitochondria or chloroplast. So like electron transport chain, the different complexes, like complex 1, 2, 3, and 4, the mitochondrial DNA will code for those proteins to be produced.
What is the endomembrane system?
Endomembrane system.
Yeah, I think it's
Yeah.
composed of like multiple like different things, like the nuclear envelope, the Golgi, lysosome. Because the main thing is like they say that eukaryotes would have like membrane-bound organelles versus prokaryotes don't have like the organelles, right? Like the membrane-bound organelles.
Yeah.
So
Sorry, yes, one more time. You're asking, what are you trying to ask?
the endomembrane system. Like what's like what defines it as like a membrane-bound organelle?
Like what defines the...
Yeah, for when comparing like eukaryotes and prokaryotes.
Okay, yeah, no, membrane-bound organelle means it
Because
has...
prokaryotes have like organelles, right?
Yeah.
So what organelles do prokaryotes have?
They have ribosomes, that's like their main one.
Okay.
Ribosomes are not like membrane-bound, right?
Okay.
It has
so,
everything.
So,
Yeah.
Yeah. So that's what I'm saying. So when it comes to like membrane-bound, like what do you mean membrane-bound?
Okay, it just means it has like an outer membrane. So like a ribosome will just look like this, whereas like a mitochondria, as we drew, where did we go? Right here, it has
Right.
like, this is like the actual mitochondria stuff, but it has, what's up?
It has inside and outside.
Yeah,
Mhm.
and this one happens to have two layers, right? It's the
Yeah.
outer and inner and outer membrane.
Right.
But that's kind of like what it means.
So then,
It's just-
for example, like peroxisomes,
Mhm.
they have like a single bilayer, right?
Yeah.
Phospholipid bilayer.
Mhm.
Okay, and then so like the other things have like, do some of them also have like two phospholipid bilayers, right?
Yeah, I
Like,
think
for example,
the-
mitochondria.
Yeah, I think it's the mitochondria nucleus that are double membrane. I
Okay.
think, but everything else that's not ribosome that the eukaryotes have are supposed to be like having at least one membrane. Did that answer your question or not?
Yeah.
Okay.
It did.
Hey, so since the
Yes.
lysosomes have a toxic internal pH, does that
Mhm.
mean they're an extremophile?
The peroxisomes themselves?
The lysosomes.
Oh, lysosomes? When we say extremophiles, we refer to organisms, not like organelles themselves. But that's actually a good thing you brought up. I forgot to mention with archaea. Archaeas are considered extremophiles for the prokaryotes because they live in like, extremely hot environments or extremely cold environments.
Yeah.
But yeah, we don't refer to actual organelles as extremophiles and stuff.
And then, for the cell
Yeah.
wall, are you gonna elaborate on that, like the bacterial cell wall?
Yeah, I'm gonna bring that up later.
Yeah.
Okay. All right, so I was talking about I don't think I wrote it down, but I don't want to erase all this so much. Everybody good with this? Can I like erase it? Okay. So yeah, there's cytoskeleton, right? Like plants have their cell wall and they still have cytoskeleton, but like they don't need it because animals on the other hand, they don't have a cell wall. So how do they get their structure? From cytoskeletons. And there's three components to a cytoskeleton, or there's 3 types of, I guess, filaments you want to call them. I think I'm going to just erase this much. Okay. So within the cytoskeleton, we have 3 types, right? The first one is, I'm going to go with thickest to thinnest. So the thickest one is microtubules. And obviously all of them deal with like structural support and all that stuff. So I'm not going to really say that it does that because that's what all of them do. But something different about microtubules is spindle fiber formation. And I'm talking about mitosis, meiosis, right? Microtubules are also where motor proteins will ride along. So I'm gonna say like site for motor proteins to move. Okay, I have to go on like a little tangent here, all right? So motor proteins, I'm talking about kinesin and dynein. This is kind of annoying. So dyneins, they'll perform something called retrograde transport. Okay, I'm gonna say a bunch of words and I'll explain it. They do retrograde transport to the minus end of a microtubule towards the cell body, okay? So let me write this out real quick. And then the kinesin will do the opposite, which is positive end, anterograde transport. So I'm gonna, okay, I'll just draw like a neuron, for example. Like here's like your cell body, and then you have like your axon going to your dendrites. And so let's say you have like a microtubule up here. The dynein is going, this is the minus end. The minus end, I don't know why, but the minus end is towards the cell body. And this is called retrograde transport, and it goes to the soma or the cell body. So dyneins will move like this way, and they'll carry stuff, whatever they're carrying, towards the cell body. Once again, this is the minus end, and it's called retrograde transport. And then kinesin will just be the opposite. It will be this way. How do you remember this? Okay, dynein, it has the word die in it, so I think of like die as something negative. So it goes to the negative end. And then retro, like if you're dead, you're old, and so that's like very retro, right? Compared to something new. Hey, whatever works, right? So dynein, negative, and retro. Another thing, negative sounds like it should be on this side, but negative is like the soma, the cell body. So you can think of like you're diving towards the cell body. You're diving towards, and so you can put all those together, right? Negative end, retrograde transport, and towards cell body. And then kinesin is just the opposite of that.
There's also, I think for dynein, there's like a mnemonic where it's like,
there might be, yeah.
dynein sounds like
Dining
dining
in a restaurant?
during like a restaurant, you know,
Yeah.
right? And then kinesin would be like carry out. So dynein would go towards the nucleus, and then kinesin would be away. Like
Yeah, I've
carried
heard something
out
like that.
from the nucleus.
Yeah, you can use that as well. I like mine because it covers all of them, you know what I'm saying?
Yeah.
Okay, next is the intermediate ones, right? They're called intermediate filaments. What the fuck? So intermediate filaments, they are, let's see, they allow for like cell to cell adhesion. And I guess these are like the most like structural based ones, like they actually do the structural things. Like microtubules and the other ones we're gonna talk about allow for actual things, whereas intermediate filaments are just there to like adhere things together and provide like structure and support. And so examples of this would be desmin and keratin. I don't know what the hell desmin is, but keratin is like, you know, protein that you found, or sorry, keratin is like what you find in your hair, right? The last one is going to be microfilaments. These are the thinnest. The main and the only microfilament I know of is actin. And the way I learned this, the person said MF actin, like mother actin, right? So I just always remember this like that, like microfilaments will be actin. And what is actin important for?
Muscles.
Yeah, what about muscles?
The thin filament.
Yeah, so they're the thin filament. She's talking about like
Contraction.
what's it called? Sarcomere stuff.
Yeah.
contraction. So actin and microfilms in general, they allow for muscle contraction. This is a much later unit, but it works with myosin and allows for muscle contraction. Some other things like calcium, as I was talking about before. And another thing that's more recent, that will be next chapter, will be cleavage furrow formation. I definitely see them asking about actin a lot, like what it does. And so no more than just muscle contraction, it's cleavage furrow formation. Cleavage furrow refers to when you're doing cytokinesis. It's like this right here, right? Like before you have like this, the two cells with two nucleus, right? And then you're trying to break them up into two distinct cells. So this cleavage furrow is being formed by actin or microfilaments. Once again, this is thick, this is thin. So there's three like main shapes of bacteria. Shapes of bacteria. So we have this right here, this, and this. The circle one or the spherical shape is called coccus. So cocci is like multiple, right? It's cocci. This rod shaped one is bacilli. And the spiral one is the easiest, it's spirali. Might be spelling it wrong. And as easy as this seems, I have seen them ask questions like, what is this shape? And it'll be like cocci or bacilli. I've never seen them ask this one because it's the easiest one, but like they could. And now we have aerobic and anaerobic bacteria. So under aerobic, there's two types. We have obligate aerobes and microaerophiles. So obligate aerobes, as it says, it's obligatory for them to be in an aerobic environment. So they have to be in like an environment that has oxygen or air. If they're not, then they die, okay? Microaerophiles are the same, they're still obligate anaerobes, they're obligate aerobes, but they don't require as much oxygen and they don't grow as much because they don't use that much oxygen. But they're still obligate aerobes, they're just a subclass of them. Now for anaerobes, we have three types. The first one is just the opposite of this one, it's obligate anaerobe, so same thing. It needs to be in an environment without air or oxygen. If it encounters oxygen, it's gone, it's dead. Second one is facultative anaerobe. This one is a little weird because they're classified as anaerobes, but they do both, right? They can grow in both anaerobic or aerobic conditions, with oxygen or without oxygen. I think they prefer anaerobic, but the nuance here is that they grow more in aerobic. You know why they grow more in aerobic conditions? Yeah, like what can you do in anaerobic conditions? Like what is the only... What? In anaerobic?
Oh, anaerobic?
Yeah.
Oh, no. Anaerobic, you could do like the lactic acid formation.
Yeah, so glycolysis and lactic acid fermentation.
Yeah.
This is all you can do, and if you spam it, you can make enough ATP, but like, you can make a lot of ATP with the oxidative phosphorylation, right? And this can only occur in aerobic conditions. So facultative anaerobes, yes, they're considered anaerobes, but they're gonna grow more in aerobic conditions. And they'll still grow in anaerobic, but just not as much. And then the last one is aerotolerant anaerobes. So I was telling you how obligate anaerobes, they have to be in anaerobic conditions, otherwise they die. Whereas aerotolerant anaerobes, if they are in aerobic conditions, they're not gonna die, but they're not gonna grow either. They're just gonna tolerate it and just chill there until they get back to their anaerobic conditions, which will allow them to actually grow. So now if I give you this tube right here, I'm gonna put, these are all bacteria at the bottom, and they're in like whatever medium, like some liquid, right? What type of aerobic or anaerobic bacteria is this? This is all I'm giving you. I see them ask this like a couple times. Facultative. Okay, that's one answer. What about you? What do you think? There's 5 options here. What about you?
I don't know.
It's all good.
So that's like bacteria?
Yeah, this is the bottom of the tube. This is bacteria down here, and this is the top of the tube.
Obviously, it's anaerobic because they're going in the bottom.
Okay. That's good. That's good. You're definitely getting, yeah.
I feel like if they had some oxygen, they would float to the top.
Mm-hmm. So which one would it be out of the 3 anaerobes?
I'm gonna go with obligate.
Oh, what did you say?
I think it's obligate.
Okay, yeah, it's obligate anaerobe. So, because look, they're all like concentrated at the bottom. What is this tube, right? This tube on the top, yo, what the? You're bugging, bro. Okay, so the top is exposed, or at least it's, you know, there's some oxygen outside. Whereas the bottom is, I mean, there might be some oxygen there, but this is the least amount of oxygen down here. If they're all concentrated here, that means that they couldn't last up here with oxygen. So they're in an area with low or no oxygen, right? And so these would be obligate anaerobes. And then let me draw the other ones too. Okay, obligate.
If
Yeah.
there's oxygen though, won't they
Yeah, that's a good question.
like die?
Yeah, I'm just saying like there might be like very little, but I'm sure like at some point there's like no oxygen.
So
It's
why,
little-
why wouldn't it be? Oh, it wouldn't be aerotolerant. It couldn't be aerotolerant because they're not.
Yeah.
Otherwise, they
I
would
feel
have.
like it would be in the middle if it was.
Yeah. Okay.
Yeah, I'm gonna draw all of them out so you can
Okay.
see.
Okay.
So obligate aerobes are gonna look the opposite. They're going to be up here. Only on the top because they are obligate aerobes. They can't survive at the bottom where it's anaerobic conditions. Let me draw this a little better. It's gonna be
They
like,
made like a race car or something.
Yeah.
wait, is that the oh, that's the courtyard.
Formula.
No, the courtyard, right?
Yeah,
That's,
yeah, yeah.
okay. I was mixing up I thought we were somewhere else, not gonna lie. So now microaerophiles are basically gonna look the same, but like there's gonna be less, and they might be a little lower because they use oxygen, but just less. But you're gonna be able to see the difference if you ever get an answer a question with answer choices. Obligate aerobes are very, very obvious. They're very prominent at the top. Microaerophiles are still at the top, but like just less amounts. This is obligate anaerobe. I'll draw one more time over here, right at the bottom. Now, facultative anaerobe. What do you think this one looks like? Based on what I was talking about earlier.
They scattered all around.
You want to draw it? Okay, so can you be more descriptive? Like just straight up scattered, or is there any concentration somewhere, or like what? It could be right. I just want to see because like there's different ways to explain that.
Honestly, just scattered all
Scattered.
around.
Because you said they reproduce better when there's oxygen present. So
Yeah.
maybe they would be more towards the top and it would be less towards the bottom.
Yeah, you got that. Good job, bro. So these are obviously going to be everywhere, but there's way more at the top, and then you're going to still have stuff down here, right? But there's a lot on the top because, as you were saying, and as I was mentioning before, they grow more in aerobic conditions, and they can do both aerobic and anaerobic respiration. So if you can do more ATP production in aerobic, obviously you're going to grow more in those conditions. Now, aerotolerant anaerobe is the one that's like all spread out, because they like obviously they grow down here, but they can like survive up here, so they just spread around. But it's gonna look like that for the most part. All right, now to elaborate on bacteria, I hate this because I had this on my MCAT, right? I didn't even think to learn this. It's very, very, very common, and people probably know this one. But I had 3 questions on this. I knew none of them. So please remember this, okay? We have gram-positive and gram-negative bacteria. Let me, I'm gonna write this in different color actually. This is extremely, extremely high yield. Why did I write these in different colors? Anybody know why? Why did I choose these 2 colors? This is-
Gram positive's purple.
Yeah.
And gram negative's pink.
Yeah, it's supposed to be pink, but I don't have that. So gram-positive, this is basic, right? I already knew this stuff, right? Gram-positive, when you do gram staining, it's gonna be purple. And then gram-negative, when you do gram staining, it's going to stain pink. The actual like color they use is called like sarferin or something. I'm just being more detailed, but it's just pink, right? Now, gram positive, they have a peptidoglycan, well, both of them do, right? Peptidoglycan cell wall. Okay, so anything I write in black is going to be something that's different, okay? So peptidoglycan cell wall plus something called lipoteichoic acid. And this peptidoglycan cell wall, it's thick, has one thick cell wall made of peptidoglycan and lipoteichoic acid. Now, gram negative, it's going to have also this, I'm gonna write PDG, okay? Peptidoglycan cell wall. But the difference here is that it is thin. And something else that the gram negative has, that gram positive doesn't have, is an outer membrane made of lipopolysaccharide. Or we also call it LPS, lipopolysaccharide. So gram positive is one thick cell wall, that's it, right? Gram negative has two layers. It has a cell wall, peptidoglycan, same thing, but it's thin. But it also has this outer lipopolysaccharide membrane. It's such basic information, but me forgetting that was very, very bad, right? Okay, I'm gonna erase this side. How do bacteria reproduce? So, what's it called? Animals and plants, or animals, right? We can do mitosis, meiosis, all that stuff. What do bacteria do? This one's very basic, you should know. Yeah, binary fission. Binary fission, it's just a type of asexual reproduction, right? Just like mitosis. Does asexual reproduction create any diversity at all? What the hell? No, sexual reproduction is what creates diversity, but asexual does not. But that's all they do in terms of reproduction. So how do bacteria gain diversity?
That's what mitochondria is too, right? Binary fission to reproduce. Pretty sure.
I'm not sure on that one.
Oh, okay.
I'll check on that. So bacterial, let's do, okay, diversity, okay? So yes, they do binary fission, but there's technically four methods that they use to get diversity, right? So the first one is going to be conjugation. There's also some high yield stuff. So conjugation, you're gonna have a bacteria like this. We're gonna call this the donor, okay? F plus is your fertility factor. So this is F plus, it's the donor. It's going to encounter a F minus recipient. And the F plus, the fertility factor, can allow for a formation of a conjugation bridge, or it's called a sex pilus. Like, you know, pili, like those organelle things. So sex pilus, this will connect, right? And it's going to donate its F plus to this one. And so now this F minus becomes a new donor. But just giving the fertility factor is not going to do anything to its genetic material, right? But here's the thing, right? This shit will keep occurring to create lots and lots of these F plus donors. And then at some point, the hope is that the F plus plasmid integrates with the actual plasmid in the bacteria, right? This is the actual genetic information. So when it integrates into the proper, genetic plasmid of the bacteria, we call this, I'm gonna draw it like this, okay? It has the thing integrated. This is called the HFR strain. I think it stands for high frequency recombination. I just made that up. But these are called HFR strains when you integrate the fertility factor into the actual genome of the bacteria. Now from here, anytime that the HFR strain bacteria conjugate with something, it can be both, like F minus and F plus. Whenever it conjugates, it's going to not only give its fertility factor, but it's also going to give pieces of its genome. So now this new genome will have new stuff in it, right? This is diverse. It's not just what it used to be, it has new stuff. And I said this can occur to both F plus and F minus bacteria. If you do it to an F minus, not only will it gain the diversity in its genome, it's going to also become F plus. If you do this to an F plus, it's just going to stay F plus, right? You can't change that, but it's going to also have the new genome. So that's conjugation. Now we have,
What
yeah.
is a plasmid that you were referring to earlier?
Like what is it?
Yeah, what is a plasmid?
It's, are you actually asking me to try it? Well, I hate this question. A plasmid is just like a, I don't know how to explain it. It's like a genetic material, but like in a circular form. I don't know like how else to explain that, but just think like it's like a circle that contains.
So
there.
for example, there's circular DNA, right?
Mhm.
So circular DNA is in bacteria and
Some viruses.
archaea, right?
Yeah.
Okay. So versus like eukarya is, you know, like your regular.
Yeah.
Okay.
Okay. So next, let's do a little transformation. Anybody remember the Griffith experiment? The something like R and S strain experiment with like bacteria. Basically, like, let's say you have like a bacteria and then it just died, right? Now it releases its DNA into the environment. You have lots of DNA in the environment. And this is from original bacteria, right? It's not just random DNA that's in the environment. It came from pre-existing bacteria. So now this live bacteria comes in, it encounters its DNA and just takes it up, and then it somehow integrates into its genome. That's called transformation. This is taking up genetic info from the environment. The third one is transposition. So this deals with things called transposons, and the enzyme is called transposase. So in the genome, let's say like your sequence looks like this, like 1, 2, 3, 4, 5, and then you have another sequence that's like A, B, C, D. Transposition will mean that you're transposing, like moving a sequence or like a piece to another sequence, right? So maybe you're moving the B and inserting it here. Now you're gonna have a new sequence that looks like this: 1, 2, 3, B, 4, 5, and then you have like A, C, D. And then obviously you change the sequence, so now something might occur, like maybe this will just stop functioning, which makes no sense, right? Maybe it will be beneficial, but I'm just saying like changing sequences can be detrimental and they can also be beneficial, or they can just do nothing. But what this does is it just allows for some diversity because you're changing up the sequences a little bit to allow for different things to occur. Now the fourth one is transduction. So transduction involves viruses. Viruses will inject into a bacteria. And what are these viruses called? Anybody know? Called bacteriophages. So transduction will look like this. Not a cyclohexane. All right, so we have this bacteria right here, and it's going to attach to the bacteria using the tail fibers. And this is the tail sheath right here, and like it will inject into the bacteria, right? So these tail fibers connect to the bacteria, but then the tail sheath comes in and injects its DNA into the bacterial genome, right? And now two types of cycles can occur here. We have lytic and lysogenic cycle. So for a lytic cycle, think of it like this. You injected that shit into the bacteria, right? So you have this and it integrates here. Now you're constantly producing, I'm just drawing these squares, diamonds, whatever you want to call them, as viruses, viral proteins. You're producing so much of them that the cell ends up lysing, right? It ends up breaking. You hijack the internal components of the bacteria, and you just produce as many viruses as you can, or the viral proteins, to the point where the cell becomes overwhelmed and just breaks and lyses. Lysogenic is a dormant version of this. It's like a precursor to lytic cycle. Lysogenic will do the same thing, right? The bacteria will have the viral, whatever, DNA integrated, but this is going to remain dormant. It's not going to produce these viral proteins for now. And anytime this bacteria reproduces, the new bacteria will still have the viral DNA integrated. And then at some point, whatever, I don't know, whenever conditions are like good for the virus, is going to activate this lytic cycle, and then this will occur, and then the bacteria will keep producing all this, and then they will all die, they will lyse. That is transduction. These are the 4 methods of bacteria's gaining diversity. Right over there. Okay, so the bacterial life cycle, when you put it into a new environment, it's gonna look like this. So I'm gonna break this off like this. All right, this is called the lag phase. This is the log phase. This is the stationary. And this is the death phase. So the lag phase is basically when you put bacteria in a new environment, they're getting used to all of the new nutrients and whatever. They're getting used to all that stuff to figure out how to reproduce and replicate there, right? So it's pretty stagnant here. Nothing is happening, no growth, no death, none of that. But after they get used to the environment, they exponentially increase. I know this looks linear, but it's supposed to be an exponential increase. So think of lag as they're lagging. They're not really doing anything. That's why they're stationary. Now the log phase, think about like logarithms, right? Exponents and shit like that. The log phase, they're growing exponentially. And then at the stationary phase right here, the death and the like growth rate is the same. And so no like net production or net growth is occurring because these are equal. And then the death phase. Death phase, why does this happen? Because the bacteria, they grow so much that they exploited, they got rid of all their resources, right? So all resources are gone, so they can't grow anymore. And after all this metabolism, they're just producing lots of toxic waste. And they're trying to metabolize this toxic waste, but it's toxic for them, they can't do anything with it. And so they die exponentially as well. Any questions on bacteria?
Do viruses fall under like the eukarya, archaea, bacteria thing, or is that just like separate?
Yeah. Okay. Yes, if viruses are considered eukaryotes, prokaryotes, archaea, any of that stuff. So eukaryotes and prokaryotes are types of cells, right? And cells are components of living things. Viruses are not made up of cells at all. They're not living. So we don't consider them anything. Eukaryotes, prokaryotes, none of that. They're just their own thing. They're viruses. They're non-living. All right. Now let's talk about viruses. Good transition, right? So obviously viruses, as I mentioned, they're not living. And they have to use like a host to replicate. And then they do like lytic and lysogenic cycle, right? Lytic and lysogenic. When we talk about lytic, quickly someone explain what was lytic cycle or whichever one, lytic, lysogenic. What is the difference?
Lysogenic is like precursor, very slow, integrates into host, just kind of sits there for like favorable conditions, and then goes into your lytic cycle, which is very rapid reproduction, just like mass produce and just like kill off your host cell and just extrude into the environment.
Yeah, good job. So lytic viruses, these are called virulent viruses, and then lysogenic is temperate. Actually know these names, okay? These show up quite often. Virulent and temperate. Virulent means lytic, like lysing the cell. They're very like virulent. Think of like viruses, they just keep growing and stuff to the point where they kill or lyse the cell. Temperate, I don't know, it's just the opposite, okay? The lysogenic one. Another thing for viruses, another like name I'm gonna mention is adenovirus. I've seen this show up multiple times. So adenovirus just refers to a virus for eukaryotes and mainly like humans and animals. So this is a eukaryotic virus, meaning a virus for eukaryotes, right? The virus cannot be eukaryotic. It's a virus that attacks eukaryotes. And yeah, once again, more specifically, humans and animals. Now, viruses, their genetic information can be single-stranded RNA, double-stranded RNA, which is really weird. When do we ever see that? Single-stranded DNA, and double-stranded DNA. So I'm going to elaborate on single-stranded RNA, right? We have positive sense single-stranded RNA and negative sense single-stranded RNA. And there's more, which I'll talk about. Positive sense single-stranded RNA, these, when they integrate into or they enter the host cell, they can automatically be used as mRNA. So automatically used as mRNA, right? What is mRNA used for? mRNA is used for making proteins. So the single-stranded RNA that's positive sense, they'll just inject into the virus or into the host, and automatically, right, they'll make the proteins, the viral proteins. Negative sense single-stranded RNA, it can't do that, right? It has to become positive sense single-stranded RNA. So how is it going to do this? Anybody know? I don't think I ever learned this. So I'm wondering if anybody remember or ever learned it. Yeah, do you know?
For
Can you increase the volume on that? The bottom one? It's like on the bottom, yeah. Yeah, what did you say on Zoom? Oh, sorry.
The replicase converts it back to cDNA, and I think it's converted back to the positive.
Do you know like the enzyme or something that's used for this?
I think a replicase. I'm not sure.
RNA replicate.
Yeah. So there's two names. There's two names for this. Both of y'all were correct. You can call it RNA replicase or RDRP. This is RNA dependent RNA polymerase. But these are both the same thing, right? So anyways, this is going to create a positive sense single-stranded RNA, and then this will be used as mRNA, right? Now, there's another thing called retroviruses. Retroviruses can only be positive sense. Yes, technically, like it would make sense for them to be negative sense, but they just don't occur. We'll talk about why. But retroviruses, right? These are positive sense. We talked about before that positive sense is automatically uses mRNA. But retroviruses, they can't do that. They have to do an extra step or two. They have to use something called reverse transcription, which involves reverse transcriptase. So we usually write RT. So I'm going to write reverse transcriptase. And this is going to convert the RNA into DNA, but we call it cDNA. So complementary DNA. So like, okay, for example, if your RNA sequence was like this, like I'm gonna put AUGC, okay? The cDNA is going to be the complement of that, which is going to be okay? And then from here, the cDNA is going to become mRNA. So we went from, instead of using the mRNA directly as, instead of using the RNA as mRNA, we had to convert it to DNA and then mRNA. That's just how these retroviruses are. And then this occurs with RNA polymerase, just using the host cell stuff. And so, look at this. This is called complementary DNA because the mRNA produced from this is going to be complementary to this, right? It's going to be the same as this. What is the complement of this? The complement of this DNA is just this mRNA, or this RNA that we used originally. So what I'm trying to say is the RNA equals the mRNA in sequence. They just have to go through an extra step to get there, to be even used. Okay, I have a question. How do we replicate, like I'm not talking about translation or anything, how would we replicate positive sense sRNA or single-stranded RNA? everybody know?
We convert it to a double strand.
Is it convert to double strand?
Yeah, I think so.
Okay, like tell me enzymes and shit like that. Like, what would we use to replicate this?
We're gonna use a replicase. I think it converts from single to double.
Okay. How many times? Once, twice, 3 times?
Easy one.
So what is using a replicase one time? What is it gonna do? We talked about how for negative sense it creates a positive sense, right? That's uses mRNA. So if it requires one to make it the opposite, what if we use replicase on this? Okay, I'm doing it backwards. Sorry. Okay. It's whatever. It's the same thing either way. So positive to negative, you need to use replicase one time, right? How do you go from negative to positive? You use replicase again. So like what you're doing is you're making a positive strand right here. You're replicating this with replicase. You're making, these are negatives, right? The black ones, and the blue one is positive. So you're making all of these negatives. And then if you do replicase again, you're going to convert these into more positive sense, right? And then let's flip it. Let's say the blues are the negative sense and the blacks are the positive sense. It's the same thing, but it's just the opposites. So you have to use RDRP or replicase twice for that. All right. So yeah, we talked about viruses. There's something, the miscellaneous section. We have, okay, these are pretty often asked prions. Hold on, let me get rid of this. All right. Prions. What are prions? We never learned this in school, but they're shown a lot in the MCAT. Prions are infectious proteins, okay? They're proteins. They're not viruses. They're not eukaryotes, prokaryotes. They're just proteins, okay? Infectious proteins that are misfolded. And what they do is, when they encounter other normally folded proteins, typically things with alpha helices, the prions cause the alpha helix proteins to misfold and become beta pleated sheets. And then when this happens, these new, originally good proteins become new prions, right? And you have a bunch of these prions just attacking these good, chilling proteins, and then they become prions, and it's just like it's a big mess, right? Prion disease is like really bad, and I don't even know how you prevent it. It's really rare, but that's what prion disease is all about. You have proteins that are just constantly being misfolded, all because of proteins. And so the main takeaway here is that they're proteins, and what it does is it converts alpha helices to beta pleated sheets, which is not what it's supposed to be, right? They were alpha helices for a reason, so they were not supposed to be beta pleated sheets, but they became that, which is bad for them. And then the last thing, another term is viroids. Viroids, these are not viruses, even though it says viro right here. Viroids are just plant pathogens. That's all I have to say about them. They're just pathogens that attack plants. All right, any questions?